I've got a smile on my face and I've got four walls around me.
Good morning. Hello. Wherever you are, I hope it's sunny. It's sunny where I am here in Guelph, and I hope you're feeling good. I hope your midterms, if you're writing other ones in other classes, I hear rumors you have other classes. I hope they've gone well and they're in the past, and that you have congratulated yourself and felt good things about all that you accomplished in all those other courses. Incredibly fast work as always on these word searches. Yeah, well done you. Let's start out with a well done you. So hands up. Pat yourself on the back. Well done you on incredibly fast word search finding. So today, with our time on this sunny Guelph Wednesday that's supposed to have a high of 7 degrees, which is going to be disastrous for our rink in the backyard if it's also sunny and hot. What we're going to do, Doug and I, is talk about annelids, segmented worms. We're going to keep talking. We talked about them already on Monday. We're going to continue talking about them on Friday. This is a super diverse group across marine, terrestrial, freshwater systems and parasitically as well. And there's some incredible adaptations to living in niches that extend from the abyssal kind of midnight zone of the ocean, from thermal vents where temperatures of seawater may reach 80 degrees Celsius, to your garden in and around southern Ontario, which until relatively recently, geologically speaking, was of course a large permanent ice sheet. And so I'm going to copy your solutions, congratulate you again on 2 very quick word, holster your stilo, and we're going to get going. Don't lose your stilo though. We're going to use it today. We're going to annotate and create some graphs together later on as we wander around the wonderful world of worms. First thing we're going to do though is talk about your skin mites, and I've got some good news about that. Jenny and Carolyn and Sherry went through all of the nasal strips that you sent back. Nasal strips. To show you. We remember the nasal strips that you slept with, and we found and we searched and found many different facial mites, and we subsampled those. We snipped them off, and we have had them being extracted and amplified for the DNA barcoding region, cytochrome C-oxidase 1, this mitochondrial little snippet that we use to provisionally identify life, animal life on the planet. And we've done that on campus at the Biodiversity Institute of Ontario, which is still working. And we've done that with the financial aid this year for your class of COESP, which is an organization in the College of Biological Sciences at the University of Guelph. And so the good news that I have is that you can see here on the left-hand side, this dark kind of array with all of these kind of little bars, little light bars. Anywhere you can see a little light bar, that means that we've successfully extracted and amplified a sequence, and hopefully the sequence of your facial mite. And that's going on right now. You can see in the white box at the bottom, some of those timestamps in the laboratory management system that they use are right up until yesterday. So that'll continue today. So that's great. I see a question in the chat. What happens if your code was never put up? So this is the unfortunate, if your code was never put up, that means that we searched your nasal strip and we did not find a mite. Sad face. So what you might be able to do, and I'm going to talk about how we analyze these, and we're going to have a big interactive kind of active session about these. But one of the things you might do is if you've got a friend in class, ask what their sample was, or just take a look at any of the other ones that'll be available, because all of these samples will be available. The anonymity is provided by the code. So whether or not you had a sample that amplified, That's, of course, not the point. I understand, as someone we've done this for 3 years now, and in year 1 and 3, I didn't have any mites, and so I felt very personally connected and kind of insulted that I was a barren wasteland for mites. So in year 2, I was very, very pleased to have a mite, and you will see pictures of it, and I will show you the sequence of it, because I have mite pride. I have mighty pride in my mighty mites. So that's coming. But enough about what's coming. Where are we? That's not where we are. Where are you at? We are at the annelids. And remember, in deep time, historically, we were taught, at least I was taught, that these annelids that we wanted to characterize, we talked about them as polychaetes, oligochaetes, and leeches. And then we understood through successive kind of exercises in phylogenetic systematics and an increasing availability of both taxa for sequencing and sequences from taxa, that in fact the leeches and earthworms are only really responsibly considered as a monophyletic group. So we started talking about the sedentaria, which has some other groups within it, but the Pogonopherons and the earthworms and the leeches are in there. Then the polychaetes with their gorgeous parapodia and nuchal organs, those diverse groups we're going to call the Errantia, or the errant worms. Now, keep letting people in here. Today, let's do this. I ended off last time with a quick description, and it's not going to let me poll. So we will come back to that. Never mind. Apparently Doug's account is taking up my capacity to give polls. But remember, we talked about this theoretical kind of arrayed, the length and the metameric segmentation for the annelid. And so we've got the prostomium at the front, and then metameric replicated segments all along, and then the pygidium at the end with the anus, and then a growth zone just in front. So the oldest segments up here by the head, and then sensory organs and concentration of nervous systems up to the end. So a real taking advantage of this bilateral symmetry and this extension along a longitudinal axis. And then we talked together about all of the benefits that metameric segmentation could provide you ecologically, and then if it was an inheritable advantage, would accrue through time evolutionarily. Now, one of the things that we talked about was, in fact, the coordinated movement because of pedal ganglia. So you're looking at parapods here with chaetae that have, in this case, in this marine worm, been adapted as protraction, and they're sticking out of those muscular parapods. Remember, the parapods, the analogy that I made was like they're paddles, and so this long extended organism, you could think of it as a many-person canoe, where the coordination of paddling is incredibly important, because otherwise you have legs bumping into each other and no successive motion. Come here. So this extends, and I think that was probably maybe intuitively appealing to you, or you could intuit that, thinking about that as a motion of a worm on the substrate, even in a marine environment. But I wanted to show you this video from the Monterey Bay Aquarium Research Institute about these other very agile marine worms that essentially fly through the water with the beating of these parapods. So the coordination of this parapod via the replicated ganglia, each parapod containing a pedal ganglia that's aligned with a ventral nerve cord, permits the coordination of the firing of each one of these legs, or paddles in this case. So today, in kind of leaving some of the talking about the polychaete, the general Errantian form, I want to talk about just a little bit to make sure that we've talked about gills and circulation and osmoregulation. Because remember, we talked about parapods, and I said one of the things that in serial homology that we could have specialization of parapods for different things. Some of them might be used for movement, where some of them might be moved for locomotion. So you can see here in this diagram from figure 14.13 of your text that many of marine polychaetes or Aurantians have gills associated with the parapods, and that that kind of takes the circulatory system out and away from the body. It increases the surface area, which is associated with the volume, is going to be otherwise a limit on the body size that these worms could eventually assume. So we've got the ventral vessel that's moving blood posteriorly, the dorsal that's moving it anteriorly, with muscular pumps and peristalsis that's moving it through, amongst and through each one of the individual segments, and then all sorts of fine capillaries out into each one of the into the parapods, into those paddles. So Rupert Fox-Barnes, again with their lovely way of recording things. So respiratory pigments, that which we're binding, the blood contains to bind to oxygen to move into oxygenate tissues. Like almost everything in polychaetes, is diverse. So there's 3 of the 4 animal respiratory pigments we have evidence for occurring within the polychaetes. So they have hemoglobin, as we do. They have chlorochuron and hemetherin. In the blood, you may see, they may occur in the blood, and they can also occur in the coelomic fluid. There's, I'm going to flip back and forth. This is your text here. This is a figure from your text. This is the comparable figure from the text that we used before. And I want to show it to you just as a small example of, there's a whole bunch of text copying that seems to me that happens as we go on through time. I would really take a look at Rupert Fox-Barnes' figure 13.25 and 14.13 from Brusca, and they're pretty darn similar. Stop. Doug, stop. There's also a neat kind of this was, if you don't follow this account on Twitter, they have really neat infographics that they produce about chemistry. And this is one they did a couple of years ago about the chemistry of different blood colors. And since we just mentioned the fact that polychaetes have many, many different respiratory pigments, both the small and the big. We'll talk about surface area and volume volume later on.
Yeah. Hello.
Oh my God. Okay, so Lily has made her appearance. Caramel should appear later on. But you can have associated with these different, so our red blood type is associated with hemoglobin. Those are ferric elements that give the red color. But you can also have blue colored blood that we see associated with some spiders, crustaceans, mollusks, octopus, and squid. That's hemocyanin. The green color that many of the segmented worms have, that's associated with the chlorochuron. And then you can even have purple blood, royal blood, if you will, the hemetherin, that other marine worms and other organisms, including brachiopods, possess. We'll talk about the distribution of the phylogenetic distribution of some of these colors later on. Stop. So in terms of dealing with metameric segmentation and excretion, this is a diagram of a simple metanephridium from a marine annelid. I'll show you it in larger context in a second, but essentially you have the ciliated nephrostome that opens into the coelom, into the inner cavity of the body, and then the nephridopore that opens to the outside of the organ. And remember, these are paired, 2 in each segment, and then along for every segment. And this was one of the advantages that we talked about in terms of metameric segmentation, is increased efficiency, that you don't have a simple metanephridium kind of trying to do all of the excretion and for the organism all at once. So in some organisms, in some marine worms, we see both proto and metanephridial systems. So the metanephridia draining the hemal system and the protonephridia draining the coelomic system are essentially just based on how large a volume that they're excreting from. But we have the circulatory pressure that we described with the heart that drives the pressure of filtration, and the nephrostome, which is here in this, so that there's the opening and out to the exterior. So this is into the coelom, non-selectively picking up coelomic fluid. The blood gets filtered across the length of this nephridium into the coelomic cavity, and then through the nephridophore to the exterior. And there's that opening when it's not carved away in this filleted annelid worm here. The reproduction, they tend to be gonochoric with some exceptions. Gonochoric meaning that there are males and females. So sexual reproduction, the gametes are stored in the coelom. They tend to have external fertilization, and they're characterized as many taxa in and around this neighborhood of the spiralia by Trochaphora larva. Now, I've mentioned the Trochaphora larva before, but this is a great place to kind of show you two pictures with this kind of furry mustache around the side. These are planktonic, long-lived larval forms that have several bands of locomotory, my emphasis is off this more-ing. So you can see here, and then the red lines align you with kind of where in the larval stage, as you move from the Trochaphora larva to what looks like a young adult, where those lines align with the prototroch is where the trochophore actually occurs. Now, in addition, many marine worms can also reproduce asexually, and sometimes even from a single segment. So you were talking about, you all correctly identified the fact that one of the advantages of serial homology is redundancy, and you keyed in on, as a group, several times the positives to being, if predated upon, they might be able to continue moving with the loss of a segment or the reduction in size of a segment. That, in some marine worms, even extends to the fact that there can be complete regrowth of this complex organism from a single segment. And it has to be not much more than, in some species, a single parapod, that very slowly that the organism can regrow from. And in fact, if you're interested, I'm not going to show this video here in class, but because of, in some marine species, the production of either sexual gametes or of reproductive forms is tied to lunar cycles and large releases of, there's all sorts of human cultures around the world that eat these things. And there's a great video about some people in the Solomon Islands catching odu, or what are also called palolo worms, that you can go and watch at your leisure. So here's my reminder to vérifier le chat, and I see I'm actually looking at the chat right now. And let's keep going. I see the question about atoques and epitoques. We're going to come back to that. Marked and noted. So within, now specifically, we're going to dive in and take a look particularly at what formerly were called the Pogonopherons. Now, this is one particular group, the Segmentation, now, that when I was taking this class, we would have characterized as another phylum, not an annelid at all. But we're going to have two kind of stories about these former Pogonopherons, one of them involving tube worms and one of them involving bone worms. So they're both deep water marine species. There's 100 plus, maybe 150 maximum number of deep water species. They tend to live in chitinous proteinaceous tubes. Some of them can be up to 3 meters long, and some of them are only kind of several centimeters long, 3 meters to 3 centimeters or into the millimeters. As adults, they have no mouth and no anus, and a lot of their metabolic needs, their nutritional needs, are met by the incorporation of chemosynthetic bacteria. These are groups that we've learned about relatively recently, and that has to do kind of secondarily with genetics, but primarily it has to do with technical advances that have allowed us to explore using remotely operated vehicles or submarines, bringing back high quality video recently and samples that have allowed us to understand and film and see this life. And we're going to share some of those surprises with you. This is one, this is the melodious tones of Attenborough talking about Pompeii worms, or the Annelids that are located on deep sea vents.
Some of the chimneys were encrusted with white tubes. The tubes were inhabited by a new species of polychaete worm that was exposed to temperatures as high as 80 degrees centigrade. No other animal on Earth was known to tolerate such high temperatures, so the scientists call these creatures Pompeii worms.
So this is kind of amazing footage, and there's really great, because lots of these initial discoveries, clearly as they're flying these ROVs underneath and exploring these thermal vents, they're recording it. And so the moment of discovery for this entire taxon is recorded, and it's largely one of astonishment, because we are kilometers underneath where any light extends. And the idea that adjacent to these thermal vents, with temperatures reaching up to 80 degrees Celsius, that there would be an explosion of life here was absolutely astonishing. It was not what the surveyors were predicting at all. Principally, the surveyors that were watching this video were geologists who were going to characterize what kind of lava forms, or et cetera, what the rocks were telling them. And then all of a sudden they had to, off the ship, get in all of the biologists to say, what are we looking at here? So in addition, so now we've got a case where that's in the kind of mid to late 20th century, 70s, 80s, 90s. Into the early 20th century, we became better at collecting these organisms at the pressure, the enormous pressures, as you can imagine, that they live at very deep water thermal vents, but bringing those up to the surface so that we can examine them in situ, or examine them not in situ, but examine them back in the lab. And since that BBC recording was made, and Attenborough talked about the 80 degrees Celsius, we've actually tested that. And so you're looking at one of these Pompeii worms here in the bottom, the screenshot of the paper, and then the adaptations that they made to their slurp gun, or their sampling apparatus on the front of the ROV, that allowed them to collect them, and then collect them in a contained at pressure environment, so that when they surfaced, they didn't just destroy the worm. And what they found, interestingly, was that the worms themselves didn't survive when exposed to the temperatures that they were measuring in situ. Does that make sense? So the temperatures, they were recording temperatures at these thermal vents of upwards of 80 degrees Celsius. Very, very hot. But when the worms were brought up and studied in a high pressure environment in the lab, any exposure to temperatures above 55 degrees Celsius proved lethal. So there's something else going on here, and that's going to require some further submarine work, perhaps some further lab work, to discover if they're in the casings that they're making, is there some absorption of that heat? I'm not personally certain if that's going to be the case, but there's something else going on, because the temperatures that they're exposed to are as high as 80, but upwards of 55, they are dead. Now, this group, the Suboglenids, continues to be interesting and continues to provide even more and more discoveries, things for us to grapple with as we continue to explore more of the deep sea environment. And this is where we're going to spend the next few minutes, is talking about Ozodax. Ozodax was discovered in the mid-80s when a remotely operated vehicle spotted a whale fall. And at the time, they thought what they were looking at was a dinosaur, this kind of long array of bones out on the bottom. And when they flew the ROV close into this decaying whale body, what they found was that it was absolutely covered in worms, and in worms of a form that people hadn't seen before. Morphologically, they looked somewhat similar to the tube worms. The females of this worm, newly discovered, were roughly the thickness of a pencil, a couple of centimeters long, and lacked mouths or stomachs or eyes. Instead, they mostly had like a large balloon-like sac which branched into this greenish root system. And that's what you're looking at over here. Now, the time series that's been playing in the top left is a view of a sampled whale fall that was discovered very early on when there was still a lot of muscle and blubber and skin that they've been tracking through time. So the worms that we're talking about weren't actually, those were, I think, hagfish, the long wiggly things. But now, here at the end of the video, these frilly bits, those are the worms, little tiny things, pencil size, 7 to 10 centimeters long. Now, so thinking about this as an opportunity, a whale, tons and tons of nutrients, bringing many, many nutrients down to the bottom of the seafloor. And that creates potentially sulfur-rich swamps or ooze that can build up around these skeletons as they slowly decay in this otherwise closely to anaerobic environment. Many organisms that they've discovered in and around these whale falls use chemotrophic bacteria to help capture the energy from the whales, just like sea vents, just like the organisms that were close to the sulfurous sea vents were doing. And so maybe it's not surprising that we see many of the same annelid groups like these subagnelids popping up here within these whale falls. So Ozodax, this worm at the bottom, has symbiotic bacteria called Oceanospirales that live inside these green root systems that help break down lipids that are present in the whale bones. So it's not actually the tissue that they're growing on. It is the bone, as the Ozodax, so bone eater is the literal breakdown of that name. So the worm has a special kind of pouch, a trophosome or a bacterocyte that contains the bacteria. They oxidize the sulfur or the sulfides and produce organic molecules by fixing carbon dioxide so that the host can use that for nutrition as an energy source. And in fact, it's not entirely clear whether that secondary use of the bacteria is what the worms are feeding on, and or if they are also feeding on the bacteria themselves. So essentially, they're chemoautotrophs, living many thousands of meters out of the range of photosynthesis. So close up, a diagram from some recent work, not a couple of years ago anymore, 10 years ago. So you're looking at kind of a cross section here. The circle is a cross section of whale bone. And then you've got a cross-section of an Ozodax growing into the whale bone to extract fats and marrow. And the two kind of branching networks that you're looking at is taking advantage of the sulfur-oxidizing symbionts and giving them the trophosome to grow within here, down within the whale bone. But then also many, many branched and opposingly laid venous and arterial systems that promote highly efficient oxygen exchange out in the frilly gills that the researchers had discovered, those kind of red branching gills that were apparent on the surface of the bone as they were flying their ROV around. So there's a whole bunch of I have many questions when I learned about Ozodax. I have many questions, and one of them is the fact that if you are, and they kind of go for this group for thermal vents as well. If you think about, it's maybe easy once you begin to understand the biology to understand how they're living where they are. But then when you think about the continuation and the connectivity of this species, if each vent and each whale fall is only a single population, how are they reproducing and sharing genes between the systems, between the vents or the whale falls? How are they finding endosymbionts? Now, what genetics has begun to show us is there's a couple of elements to that. One of them is that the Oceanospirales, for the Ozodax in particular, that bacteria that the Ozodax requires lives it can live freely in the water. So there are some, and in fact, once the worm itself dies and the trophosome breaks open, the symbionts are released into the environment. So that long-lived Trochaphora larva that we talked about earlier as characterizing the Arantians, the polychaetes, is really important for floating around in the environment, drifting through currents, and feeding and potentially then connecting otherwise reproductively isolated populations of individual vents or individual whale falls, but also for running in and becoming infected with the required endosymbionts, the bacteria that are going to let them live as chemoautotrophs in these deep water environments. And I love, there's going to be so much that we have yet to learn about the co-phylogenetic relationships of these bacteria and their hosts. It's going to involve currents and marine biology and understanding of ocean currents, and the logistics just make me quite excited. So I hope that some of that excitement is shared with you. And one of the things that I'd like you to do now to use the chat is think about this figure here. This is from one of the papers that I just presented. To think about how Ozodax worms change the faunal, do they change the faunal communities of the whale fall? So on the left, we have vertebra, single vertebra, or two of them here, paired vertebra of whale vertebra that have not been colonized with Ozodax. And then take a look at the appearance of the whale vertebra here on the right. And what I'd like you to do is to jump into the chat and tell me about kind of functionally, give me some ideas about functionally what is Ozodax doing to the bones? What do you think is happening so that we have a change in the community structure, visual change anyhow, between here on the left and here on the right in A and B? Give me your thoughts. What are the worms doing to the bones? What are the worms doing to the bones? What do we got? Oh, I like. Okay, let me arrange my screens here. So we have hypotheses, a couple of them. Breaking down the vertebrate and the bones so that the nutrients can be more easily absorbed and taken up by, presumably by different species. I like that idea, SCARS-02. The dash, breaking down the bones, releasing nutrients for more organisms, thus increasing diversity. Opening the hard outside of the bone for other organisms. I like that hypothesis too. They could be changing the medium so that the bacteria no longer thrive. Maybe these bacteria can grow only in concentrations. Yes, and I like that idea a lot. So there's a whole bunch. So there's kind of two things. There's that I'm seeing already in your ideas. One of them is changing the physical structure of the bone and breaking it down, and the other is changing the chemistry of what's potentially available for other organisms, other taxa to take advantage of, leaving more room for other organisms. That's fantastic. And both of these are largely correct. So put your hand up. Doug, do not eat my book. So as a word about your text that Doug just had in his mouth, and a nod to the fact that sometimes going to school is hard, even if you're a collie. This is Doug eating some data of mine from our last lecture that did not all go all the way through. He just nibbled it, but I was asked what data was on this card, and my response is it doesn't matter because it's gone. But watch collies. Yeah, Doug wants to learn. He wants to really absorb. So this is kind of one look. This is another look at from the vertebrate on the left here from the same study that the photo came from. So there's they've characterized these bacterial mats on vertebra. That's what I've traced in red. And then vertebra 1, 2, 3, 4, 5 that have a bunch of different taxa, but no Ozodax. And then you see here 6, 7, and 8, and 9. That's where the Ozodax colonized, and what they saw immediately was a change, a reduction in the bacterial mats, an increase. That's this dashed line in the degraded area. So that's kind of, you've come up with it already. It's changed the chemistry and it's changed the physical nature of it, and that's permitted two things. So it's degrading the bone, it's allowing water to diffuse through matrices of the bone, bringing more oxygen into that matrix, and so the sulfur bacterial mat community is reduced, and then there's a lot of other kind of secondary facilitation that happens. So well done, you did the work of this paper all at once, sitting wherever you're sitting, presumably in comfy pajamas. Or comfy clothes at any rate. So right hand up in the air, pat yourself on the back. Well done, you deep sea remotely operated vehicle Ozodexologists. And as another trend that you're going to see, that you have experienced with me throughout the day, is that we are still discovering more and more species of these things all of the time. So that's core elements of their basic biology of some taxa within this large family, or this family that was formerly an entire phylum of its own. And so, of course, when we find whale falls, as you might imagine, in different parts of the world, much of the work in the last was done off the coasts of California. On the other side of the Pacific, or in this case, rather in the South Atlantic, new whale falls, new species. And so there's, like I said, there's the logistics of this are writ large as phylogeography. So the geography of phylogeny is fascinating to me, because there must be connectivity. Every whale fall can't be a new species. That just doesn't make sense in any evolutionary way. So there has to be connectivity. However, there's such isolation associated with thermal vents and whale fall that there's probably an enormous amount of diversity here that we have no idea about, just because it's very diffusely arrayed across the bottom of the ocean. Oh, that's awesome. Sherry just put in the chat that Ripley's, not Riggs, Ripley's is having a whale fall exhibit soon. So sign me up. Let's get a bus and we'll go magic school bus it the whole way, all as a group. I am already in the chat, so I verify. So Doug and I will continue. And we're going to move from the marine environment now into the terrestrial environment. And we're going to talk about the Clithalata, formerly called the Oligarchs, so earthworms containing cocoons, no appendages, and clitellum. So apomorphies for this group, that does also include the leeches, is that they lack parapods, so those muscular kind of paddles, they're not there, and they have a clitellum. So this is a series where some of the, and remember serial homology, that there can be specialization of some of these segments. So here there are some of the anterior, some of the oldest segments become enclosed in a thick glandular epidermis that we call a girdle or a saddle that becomes a reproductive structure. I see a question in the chat just as we're leaving here about the whale fall. How long? It can take years. How long does it take for a whale to break down? Years and years and years. So musculature within the oligarchs is similar to polychaetes, but it's reduced because it's not, we don't have the parapods, but we see the same, we see retractor muscles, longitudinal muscles, circular muscles, ketal pro and retractor muscles. And so the chetae is still there, and there's still traction provided by these chetae that you'll see or have seen in moving of peristaltic kind of moving of worms. But it's reduced. So at a distance with your binoculars, this is a worm that doesn't apparently have feet or paddles. So we've talked about the hydrostatic skeleton before. We've talked about the And I've shown you actually some earthworm video, even though we were talking, I believe, about rotifers at the time, to think about the antagonistic firing of longitudinal and circular muscles to, in broad strokes, talk about how you might move with a hydrostatic skeleton. So you can see here in this diagram, so the animal moving, so projecting, so if we start at C at the bottom, but say we've got these firing of alternating action of circular and longitudinal muscles. So from A to C, the contraction of the circular muscles here, from anterior to posterior, and then that coelom is then kind of pushed, that hydrostat is pushed forward, and that's facilitated by these cheetae. And you can see the cheetae shown in these diagrams associated with those segment numbers, and those are facilitating the anchoring of the worm so that it's not slipping through the substrate as it's moving along. Then in D to F, the bottom part of the figure where we've got, and this is the peristaltic part, this rhythmic wave of action, is that once it's been shot forward, it needs to then fire longitudinal muscles, and so that we've got essentially an anchor set up at the front, so that once you've projected that part of your body forward, you need to expand the anchor, embed the cheetae into the substrate so that your peristaltic action can help inch you along. And that's the facilitated, I hope, by the video that's playing in the bottom right-hand corner, visualizing that. The nervous system for the sedentaria or the oligochaetes, the brain is slightly more posterior than it is in the that we saw in the Orantians and the polychaetes. They have a single giant ventral nerve cord, again with some giant axons that we saw. No eyes per se, like we saw with some of those highly mobile polychaetes, but definitely photoreceptors that are distributed around the body. So becoming negatively phototactic is super important. So we see segmental nerves and coordination by this giant single ventral nerve cord here. They have a muscular pharynx. And this is associated with, as you can imagine, if you're picturing for this part a terrestrial earthworm, that's kind of where to go with a lot of these diagrams. That's where they've come from. So that's breaking up. That pharynx is helping to break out the largely just soil that they're pulling into their mouth, breaking it up. And then the esophagus is quite complicated and long and chitinous with chitinous projections that help grinding. They have a crop here. So the gizzard here and the crop slightly anterior to it. And then they're to extract this kind of their needed nutrition from this complex matrix of soil, of abiotic kind of inorganic material. You can imagine that facilitating that process is going to be involve a change in the surface area of the gut, that they're going to need to increase the surface area of the gut. And that's what we think this, what we've called a typhocele is here. So that's the fact that the gut is not just this kind of a cylindrous, an open cylinder tube, but in fact it's folded back on itself as a U that helps increase the surface area and then increase the amount of absorption that can happen from what might be a fairly dilute tissue source. The Chlorgonic Tissue is the center of glycogen and fat synthesis. And you can see it kind of lining that fatty-like drawing that's lining the U-shaped gut. Their circulation, their vasculature is similar to polychaetes that we saw already. They tend to just have hemoglobin, so not as much diversity in respiratory pigments. They tend also to be fairly resistant to hypoxia, but you can see circumesophageal vesicles in the last diagram. And then for excretion, this is a look at a single Nephrodium. You saw a similar diagram. It's a little bit more complicated here, associated with living in a largely terrestrial environment. But essentially, these the nephrostome is acting in the same way that for each. So this is the septum that's dividing each one of those metameric segmentations. The nephrostome draws the coelomic fluid through, and then as that material migrates along the narrow tubes and then the wide tubes, that there's a concentration, and that different nitrogenous wastes are picked up, and then there's selective reabsorption in the blood and leaving essentially a urine that's rich only in those nitrogenous wastes that is then exited from the body. Their sex involves the clitellum, and I'm going to do a bit of a time check here just to make sure. Yeah, we can. For those of you who are joining us asynchronously, it's quite likely that the BBC will ask us to strike this part from the video. So make sure you go to the YouTube link that is here and also in the chat and in the PDF to take a look at what we're going to share right now, which is David Attenborough. Beautiful. If you haven't seen Life in the Undergrowth, you're in for a treat. Try and find it on the interbyte somewhere. I'm not sure if it's on Netflix or not, but it's gorgeous. So, oligocete reproduction. Those of you who have already had your lab have probably already seen this, I think, with Sherry, and that's Isabella Rossellini. In the gift to the world from European acting circles, playing an earthworm involved in reproducing. There's simultaneous hermaphrodites, copulation involving the exchange of sperm, and then the clitellum, that kind of girdle that encircles these anterior segments, is involved in creating the egg case, and then the egg and the sperm are injected into that egg case. So here is a look at some of, in the bottom left-hand side of the screen, these diagrams are about kind of showing how we use the clitellum as an identification tool for different species of oligochaetes or for earthworms. And because sometimes their formation varies between different species or between different taxa. One of the cool things to me about earthworms, it might have been, for me, it may have been one of the first scientific epithets or scientific names that I was ever asked to memorize, Lumbricus terrestris. Maybe it was for you as well. And what I find neat is the fact that genetics, very simple genetics, just in fact, in this case, in this study from 2010 that Sam James did with colleagues around the world, I think I know 4 of the 2/3 of that author list have been in this house. So that's kind of cool. But they discovered that Lumbricus terrestris is not actually, this model system for many, many things is not actually just one species. It's more than one species. And that's a concern if you're, for example, a physiologist who are using any kind of model taxa as a way to understand a physiological process. If your results, your experimental study may have in itself variability and variation, but if some of that variability is occurring because within your study you have more than one species, that makes your conclusions difficult to depend on, and it certainly makes your results very challenging to compare across multiple studies that might be trying to move science forward modularly. So this was an important study that they did a few years ago to help rediscover the type, specimen, which you know about from your naming experiment or your naming assignment. But also the fact that there is more than one. Lumbricus terrestris is not just one thing. There's another closely related lumbricus called Herculeanus that they have you can see in this name. This was proposed more than 100 years ago, but then in the intervening time had been synonymized, but they have rebirthed. Now, in the last minute, I'm going to kind of set the stage for what we're going to do. We're going to start with a bit of a labeling experiments and some graphing on Friday. One of the things I've called attention to a couple of times is that the earthworms that you have experienced or shared, if you're in Ontario, if you're in Canada, if you're in much of the Northern Hemisphere, the earthworms that you share the place with aren't likely endemic, that they're invasive and they've been moved, in our case here in Ontario, largely by European colonizers. And that changes the environment dramatically because the forests that are here that predate the worms, they're consuming leaf litter and they're changing the understory of the forest dramatically, changing the physical and chemical properties of the soil, and that's adversely affecting many native species and is going to change the natural functioning of forest ecosystems that you experience and likely in your lifetime. And this migration is still happening. Worms are arriving in and around the lower southern edge of the boreal right now. There are worms in Algonquin Park that were not there a decade ago. So as you kind of revisit your notes before Friday, I'm going to present you with some data about how we might actually be measuring and understanding these historical physical changes that earthworms are causing. So in the intervening time, between now and Friday, enjoy your day. Take advantage of the sunshine. Be good to yourself. Be good to the people that you're around. And we will see you. I'm going to be here in the chat. I saw there was a couple of questions that I didn't get to answer, so I'm going to answer those straight away as I transfer the PDF and the video up into the YouTubes. But other than that, I will see you Friday. Ta-ta. Doug says goodbye also. Cool.